Moore Medal as the premier recognition for early career success in innovation, as reflected in both market impact and improvement to the quality of life. By highlighting extraordinary individuals and their work, SCI America aims to promote public understanding of research and development in the modern chemical industries, enhance the interest of students in applied chemistry by providing role models, and emphasize the role of creative research in the global economy. Based on the criteria that the award name draws attention to young innovators and connects to the established Perkin Medal, which will be given tonight, the Executive Committee has decided to name the award for Gordon M. Moore, co-founder of Intel and a seminal figure in the establishment and development of the semiconductor industry. Mr. Moore has often described Intel as a chemical company. As a research chemist and then director of R&D, Gordon Moore carried out pioneering work on silicon transistors, the integrated circuit, semiconductor computer memory, and the microprocessor, and all this while well under the age of 45. Well, history speaks for itself sometimes. Many great achievements have been done at a young age, and this is also characteristics of all the awardees of the Moore Medal. And it's now my great pleasure to introduce Kathleen Shelton, Director of Technology of DuPont's crop protective business, to introduce Andrew Taggi. Please, Kathleen.
Thank you, Carsten. Let me start by thanking the chemical industry, the Society of Chemical Industry, for their support of the Gordon E. Moore Medal, and the Chemical Heritage Foundation for hosting Innovation Day and this luncheon. What a lovely event we're all able to participate in today. I'm really pleased to be able to introduce Drew Taggi to all of you. He's someone that we're extremely proud of here in DuPont, and someone that I know all of you will learn a lot from in his presentation coming forward. You know, the Food and Agriculture Organization of the United Nations has estimated that over 840 million people in the world are malnourished or undernourished. And one of the key missions of the crop protection business in DuPont is to provide the food, the fiber, the feed to meet the needs of our customers, but of society, and hoping to decrease this tremendous problem that we have around the world. Drew will talk a little bit about his work, and I hope many of you will see the impact of that in this worldwide problem that we have. We're fortunate not only to have Drew as a contributor to our scientific community, but his father as well, who contributed for many years in his career at DuPont, and we welcome him and his wife Alicia here today, as well as Drew's wife Suzanne. So thank you for joining us, as well as some other friends and family of Drew's community. So Drew began his scientific tenure when he started his undergraduate work at Cornell, and then he moved on to do his graduate work at Johns Hopkins University. He then returned back to Cornell as a postdoctoral fellow, and then joined us here at DuPont. He joined us in 2004 and is now part of the Discovery R&D effort. He's worked in a couple different areas, most notably in the herbicide discovery as well as the fungicide. You know, the agricultural community requires new products because resistance and new mechanisms of action are always necessary in order to be able to contribute to the pest issues that we see. So we're always looking for novel biochemical modes of action, as well as meeting the consumer's needs or the grower's needs around having products that are more sustainable than the ones we may have on the market today. So Drew is being recognized for his significant contributions to a discovery of a new broad-spectrum fungicide, which possesses a brand new mode of action. Discovery chemists don't ever really get to work alone. They work within multidisciplinary teams. And so their challenges are not only to create new molecules with novel modes of action, but to also have them meet certain toxicological criteria, efficacy, environmental fate, physical properties, so you can begin to understand the tremendous challenge it is to bring a new product or a new molecule to the market. Drew works in these multidisciplinary teams to try and bring these products to market in a way that they can meet or exceed the needs of our customers. And sometimes it's just hard work. Drew and his team have synthesized over 1,500 novel compounds to refine the targets in which we are now looking at some molecules that we believe will be commercialized in the next couple years. We have found a molecule that will meet all the requirements for a broad-spectrum fungicide, and in 2019, we hope to bring this product to the market. I'm sure Drew will tell you more and get you excited about this product as we are. So let me join all of you in welcoming Drew to talk about his work and his accomplishments.
Before we have the joy to hear his talk, Drew has the joy to receive this medal, which I just hold here. I hope it doesn't fall down. It's my great joy and honor to give you the 2014 Golden Moore Medal. Congratulations. Congratulations, Drew. So some pictures. Yeah.
Well, thank you, Kathy, for the kind introduction, and I must extend my sincerest gratitude to the Society of Chemical Industry, the Chemical Heritage Foundation, as well as the Selection Committee for bestowing upon me this great honor. And when Ron called me up this past winter, I was, of course, extremely excited about winning the Moore Medal. I was also very excited about the attention this would draw to the wonderful team we have at DuPont Crop Protection, and also the opportunity to come and talk to you about chemistry and agriculture. And before I started in crop protection, I really knew nothing about the crop protection industry and how chemistry was used in agriculture. It's not really taught at the university level, and so I thought this would be a great opportunity to tell you about our motivations, the challenges we encounter on a daily basis, and as well as how someone ends up in crop protection. Because it's not a university subject, we all come from really a diverse set of backgrounds and all end up doing the same thing. And so I started out in my graduate research, and I was working on catalytic asymmetric synthetic methodologies. And so we were able to develop a synthesis of beta-lactams and high enantiomer diastereomer selectivity. We did this by overcoming the inherent instabilities of monoketones, and by using an organocatalyst, we're able to impart very high selectivities. And then subsequent to this, we were able to extend the methodology to prepare alpha halo esters. And so these are important synthons to the synthesis of other materials. And so when we were finishing off this work, and I was thinking about doing postdoctoral studies, my advisor, Tom Lechta, as well as Professor Gary Posner, who was on my committee, they both suggested that I go off and do something completely different. And Gary even went one step further, and he suggested that I go and do something fun. And really, because he kind of told me this was the last time I was going to be allowed to have some fun. And so I had signals my intention to go into industry, and so maybe this was a sign that, at some point I was going to become a working stiff and never have fun again. And so I took all their suggestions to heart, and I returned to my alma mater and joined the research lab of Professor Jerry Meinwald. And so Jerry had been in decades-long collaboration with Professor Tom Eisner out of the entomology and neurobiology departments. And so together they had really founded the field of chemical ecology. And chemical ecology takes natural products one step further. It's not just the discovery of the molecules, it's the discovery of the purpose of the molecules and really how chemistry interacts in nature. And so one of the projects that I was really excited to get involved in was looking at the small molecule constituents of spider venom. And so we started out by looking and really screening over 100 spider venoms just using proton NMR of the unpurified venom extract. And very rapidly, we realized that you could actually learn a lot just by looking at the secondary metabolites of the spider. And so if you gave us just a 1D proton NMR, you could tell at that point whether the spider was a hunting spider, an ambush predator, whether it was a web weaver, whether it was an archaic spider, a more modern spider. And one we got really interested in was Tegenaria agrestis, known as the hobo spider in the western United States. And just from that initial proton NMR, we were able to see that there was going to be something interesting. And so using multidimensional NMR spectroscopy of the unpurified venoms, we were able to discover this family of sulfated and glycosylated nucleosides, which hadn't been described before. And really thought it was the methodology used and the fact that we weren't purifying the venoms that led us to this discovery. Because these molecules, once separated apart, actually tended to be fairly unstable. And so it was only when they were together that they would really exist. And so in addition to the menagerie of spiders that I kept in the Eisner lab, I tried to get involved in as many other projects as possible. And so we worked on diurnal fireflies. So these are fireflies that had moved from the nighttime into the day. They you fly really slowly. I mean, you know, as a kid, it's like it's one of the easiest things to catch is a firefly, and you might ask yourself why nature does that. It seems to be a counterintuitive thing, but it turns out fireflies are actually wildly toxic. And so if you were to catch one and feed it to your pet lizard, the lizard would die rather rapidly. And so they really live a carefree life because nothing will eat them. Got into other defensive chemicals, so caterpillars and other larvae. And then remember at one point I was chatting with Professor Eisner, and he started telling me about a species of invasive earwig that had gotten a foothold in the New York ecosystem. And he was really interested in understanding how they did this. And he thought, potentially defensive chemistry had played a role. And for some reason, the biologist didn't want to go collect them. And so he needed to dispatch me to do this. And in retrospect, this might have been some level of biologist-chemist hazing, because actually the picture on the lower right, this is it turns out the earwigs lived in the manure pit of a Wegmans egg farm. And so it's hard to tell from the scale here, but those are 100-yard long, 5-foot tall piles of chicken manure. And there were 20,000 chickens above, and everything was raining down. And sure enough, there were earwigs in there. It reminded me very much of when I was growing up, Indiana Jones and the Temple of Doom. And when you just kind of stick your shovel into one of these piles, the insects poured out like water. And so we collected the insects, and I worked with Eisner on this, and turned out it was defensive chemistry. And it really just, all the projects together really instilled for me a love of biology and chemistry on the interface of biology. And really gave me that, like my first taste of field biology and getting out of the synthetic laboratory. And so when the opportunity at DuPont came along, I thought this might actually be a chance for me to continue doing what I was doing, to continue having fun, even though I was promised that would stop, and move on to look at a new set of challenges at the interface of chemistry and biology. And so we moved on and started working on what is potentially one of the greatest challenges of our generation, and that's food security. So how do you produce enough food and enough quality food to feed the world? And so when you start looking at the numbers, it's actually quite shocking. So in 6 out of the last 11 years, we've consumed more than we've produced. So, what does this mean? Well, 10 years ago, the world grain stores contained enough food to feed the world for 150 days. And now those stores have less than 50 days of food. And so you can tell the system is it's very strained. So one way to deal with this, of course, would just be to plant more acres. And while this is a potential solution, it actually has its own consequences. And so for every acre that you plant, it's another acre of biological diversity that you use or that you lose. And so the alternative is to really maximize production from all the acres that already exist. And so if you had a way to address pests, you could potentially recover 20 to 40% of food production every year. And so these pests we're talking about are things like fungi and bacteria, weeds, nematodes, and insects. And so by dealing with those, we potentially have a solution to at least part of the food crisis. And so these ideas are not new. Actually, Norman Borlaug, who got his start at DuPont Central Research, had pioneered many of these ideas back when the world's population was only 2 billion. And as we've crossed 7 billion and on our way to 9 billion, you can see how the problem has only gotten worse. And additionally, things like globalization and changes in weather patterns have really kind of brought their own problems to us. For instance, a good example comes out of Europe. And so prior to the 90s, Septoria tritici was not really an interesting pathogen. It was there, but really the major concern were the pests that Borlaug was interested in, leaf rust and stem rust. But in the 90s up until now, this pathogen has really exploded and ultimately can cost between 35 and 50% of yield out of European cereals. And then if you look to South America, prior to 2001, Asian soy rust did not exist. It's thought that the spores actually blew across from Africa in a particularly strong hurricane season. They quickly colonized Brazil, and this is an extremely vicious disease with yield losses in the 50 to 75% range. But something that we can all, we've all really experienced on the East Coast is actually the marmorated stink bug. And so prior to '98, this didn't exist to us. And now it seems like any warm day in the winter, they're flying around your house. And they're not only a nuisance to homeowners, but they are an agronomic pest. They will eat crops. But also, like many insects, they're a vector for other disease. And so, for instance, I had peach trees in my backyard, and when the stink bugs came on the scene, they would suck the juices out of the peach, which, overall, it didn't leave, much damage, but that wound is a great starting point for a botrytis infection. And so instead of having nice orange peaches at the end of the season, I had these big gray, hairy peaches. And, it just, not really good for me. And at this point, it's not known what the economic impact of this insect will be, and, only time will tell. But so you can see how the system is really strained and challenged, and there's been times in the past where the strain system has broken entirely. And so some really good examples of this are things like the Irish potato famine. So this was caused by potato late blight on potatoes, and it resulted in the death of 1 million people and another 1 million people leaving Ireland. So overall, 25% of the population disappeared from that country due to one pathogen on one crop. And then 100 years later, the same story repeated itself in Bengal, where over 3 million people died from starvation from a single pathogen on a single crop. And so one possible way to deal with this is actually through chemistry. And so chemistry has a long history in agriculture. So it dates back to 2500 BC when sulfur was discovered as a fungicide. And it's still used today in vineyards throughout the world. And then you moved into the heavy hitters, the arsenics, mercury, and lead, and then luckily onto the organic molecules, things like nicotine, pyrethrum, and rotenone, all insecticides. And then in the 20th century, this was really the big explosion for crop protection in both the number of new chemistries as well as the different modes of action, and things like 2,4-D, glyphosate, the sulfonyl urea herbicides, also discovered at DuPont, really led to a landmark in use rate. So where you may have needed kilograms of compound, instead 15 grams is all that you had to. And so DuPont got involved in crop protection over 100 years ago. And so this is actually a brochure that DuPont put out to growers advocating the use of dynamite in agriculture. And the benefits that they talk about are still the same benefits we're looking to today. So saving time, money, labor. It's estimated that if you remove herbicides from agricultural production in India, it would take approximately 4 billion person days to do the weeding, and so to replace what a chemical is doing. The punchline, of course, was bigger yields, and that's still what we're looking for today. Growers 100 years ago and growers today want to get more out of the acres that they have. And so what are we looking for now? And we're not looking for explosives, and we're not looking for heavy metals. We're looking for compounds with extremely high safety margins, so things that are non-toxic to non-target organisms like mammals, birds, fish, beneficial insects like honeybees. We also want to be extremely effective at very low use rates with a broad spectrum of activity. So this way you're putting the smallest amount of compound in the environment as well as the smallest number of compounds into the environment. You also want to be compatible with integrated pest management. So if you have ladybugs eating aphids, you don't want to kill the ladybug with your insecticide. And then finally, as Kathy mentioned, new biochemical modes of action. And so nature is always reinventing itself and presenting us with new challenges or old challenges. And so potato late blight, which caused the Irish potato famine, is still the #1 problem in global potato production. And that's because of resistance and nature just kind of resetting the clock. And so we're always having to go back and reconfront these challenges. And so we're working at the interface of chemistry and biology and really looking for a sustainable solution and feel obligated to do things better and safer and greener than we had ever done before. And so we think about all these things when we start out into a new area of chemistry. And so you can start out potentially at the target. And in order to achieve that broad spectrum of activity and potency, you really need to attack a well-conserved pathway. but at the same time, that pathway may be well conserved in off-target organisms. So you need to exploit subtle differences in active sites, metabolism, and bioavailability in order to achieve the high safety margins that are required by regulatory agencies. And so there's a lot of crossover between pharmaceuticals and agrochemistry, and a lot of their target sites are the same ones that we would be interested in, and so we can take inspiration from each other. And you might target things like amino acid biosynthesis, respiration is a great thing to look at, or transcription. And a good example of some of the inspiration between the two industries is actually Zyvox, which bounced back and forth between herbicide discovery and pharmaceuticals before eventually making it as a human antibacterial. Where it really diverges from the pharmaceutical industry is when you get to thinking about the patient, if you will. And so the pharmaceutical industry has spent hundreds of billions of dollars understanding metabolism and translation in a single organism. And in crop protection, we're confronted with hundreds of organisms and then in a very complex environment. And so things that wouldn't be much of a concern, a little bit of photoinstability in pharmaceuticals could be spun as a good thing. For us, it could be catastrophic. All the compounds have to be formulated and in many cases sprayed onto plants. And that formulation is an area you could be studied as a PhD project in physics. When you change the molecule, you change what sort of formulation solvents. And that affects the type of droplet. Does the droplet stick to the plant? Does it shatter? Does it roll? Does it penetrate into the plant and lead to curative activity and systemic movement? These are all things that we need to understand. Then what happens when it rains? Does the compound wash off the plant? Clearly this is a problem for efficacy, but also it's a problem if there's runoff into a stream or a lake. So how does your compound interact with the aquatic organisms, let alone the beneficials that may have been living on the plants that were sprayed? And then as the compound moves into the soil, you have a whole additional set of organisms. And you need to understand what is the ultimate fate of that compound. So does it degrade biologically? Does it degrade chemically? What metabolites are formed and where do they go? So in Europe, there's a very strict cutoff for groundwater concentration. If you crossed 0.1 parts per billion concentration, you can't get registered. And so this is something that we're keenly aware of and we have to keep a close eye on. And then finally, you do have to think about the end use of the crop. And so it's known classes of fungicides like the strobilurins, not only do they kill fungi, but they actually have a physiological effect on the plant, which results in a yield boost. But when you get this yield boost, does the balance of protein and carbohydrate in the grain change? What happens when you make bread? What happens if you're making wine from grapes? Does the wine taste differently after it's been treated with one fungicide or another? These are all things that need to be dealt with. And then finally, even the issue of silage. So you might take the grain and use it to make bread, but where does the straw go? Do animals eat that? And what's the ultimate fate of any residues that might be on there? And so it's an extremely complex situation that needs to be tackled. And we try to tune as many of these different attributes into the actual molecule. And each one of these attributes represents an entire team of people. And you can make very subtle changes in the molecule and they can have huge effects. Recently we saw in the field, just changing a fluorine to a chlorine, it changed the melting point of the molecule by 50 degrees. Solubility and formulation solvents changed drastically, and all of a sudden the activity in the field shot up. We started seeing beneficial plant effects, curative activity, systemic movement, all because a fluorine changed to a chlorine. And so it's really important to understand each portion of your molecule and what it's there for and optimize it to all these different attributes. And so we are advantaged in crop protection in that we can jump right into in vivo activity. And so if you want to look for herbicides, even in a high throughput manner, you could run a 96-well plate with broadleaf weeds. You could use a camera to determine the percent green leaf area, whether or not you have a good herbicide or a bad. You can carry the same concept over to insecticides. If you dye the food orange, and then if the insect is either deterred by the molecule in that food or killed by it, you have an insecticide hit. But as rapidly as possible, we want to get to the real situation. And so we'll quickly turn to larger plants, and this would be an example from fungicide discovery where we're screening on grapes and, on grapes and wheat, tomatoes and rice, and then these plants would be subsequently inoculated with a broad spectrum of pathogens so we can start getting our first idea of potency and spectrum of our molecule. And so An example that I got involved in very early on in my time at DuPont was actually, really when I walked in the door, was an area of tubulin modulating fungicides. And so prior to our work at DuPont, there had been 15 or 20 years of other industrial activity looking at these compounds. And while there had been many exquisitely active compounds made, none of them had all the attributes necessary to make it to the commercial market. And so we were interested in building off of some art from BASF, which was recently published. And the idea was, very simple, very subtle, but could have broad impacts in the molecule, where we take a ring nitrogen from the pyrimidine and we change that into a carbonyl. And so if that nitrogen, say, is important in binding, potentially the lone pair on the nitrogen can be mimicked by the lone pair on the oxygen. We have these pyrazonone fungicide targets, and luckily for us, they were extremely easy to make, and so we were able to get them into our fungicide screens rapidly. And so at the time, our fungicide screens were focused in heavily on the European cereals market. And so we had 4 pathogens of wheat, including Septoria tritici, which was the new problem, and then Borlaug's old nemesis, wheat brown rust. In addition to that, we looked at specialties, things like Rhizoctonia on turf, botrytis on tomatoes, anthracnose on cucumbers. And what we found was with all our initial analogs, we saw really excellent potency as well as spectrum. And so this got us extremely excited, but right off the bat, we knew from the chemistry lab that there was a concern. And so when you synthesize these molecules and you look at them on TLC, under a UV lamp, you see a big purple spot. And that was a concern from us right off the bat, because that means we were absorbing long-wave UV. And so sure enough, if you look at the UV spectrum of one of these molecules, you see this absorption peaking out around 370. And so that's a concern, but it's not the end of the road, because even if the molecule absorbs light, if it doesn't have a degradation pathway where the molecule will fall apart, it's really no big deal. It can go back to its ground state. And even if both the absorption and the degradation pathway are present, all the compounds will be sprayed onto plants. If the compound penetrates into the plant, the UV defenses of that plant can actually protect the molecule. So the real test that you need to look at is a foliar photolysis experiment. And so in this case, we looked at wheat leaves, simulated sunlight, and following by LCMS, we started seeing the appearance of a molecular ion at 214. And for us, this was very bad news. It came in very quickly, and that molecular ion represented the upper right-hand quadrant of the molecule. And so it turns out that core that we had, the change we had made, while it gave us exquisite biological activity, was also the Achilles heel for our area, and really forced to go back to the starting point. And so, but because we knew that our problem was with the core and with UV absorption, it gave us kind of some direction. And so we spent a lot of effort, and a lot of chemists were involved in looking at different cores that we could replace the pyrazonone with. One idea would be to actually remove the carbonyl we had installed and go from a 6-membered ring to a 5-membered ring. You'd want to keep all the pendant functionalities the same, because when you go back and look in the literature, all those groups were always present, and it looked like we could just change the core and go from there. And so the synthesis of these was straightforward, and it was even easier if we actually switched the N substituent to an aryl group, and that would actually enable us to use some different chemistry, where we do a Staudinger reaction, cyclize with Phillips-Myers reagent, and then just halogenate and then displace with a pyrazole. And so we could quickly get to our desired targets. And when we compared these with the pyrazonones, unfortunately what we saw was, while we were photostable, the spectrum and the potency was gone. And so you might wonder, why this is. I mean, we have all the right groups from all the previous molecules. What led to this big change? And if you look at some molecular modeling and some 3D overlaps, you start to see, while we do have really excellent overlap on most of the molecule, those pyrazoles are no longer in the right place. And so one way to deal with this would actually be to add a linker. And if you add that linker and then look at your models again, now you have overlap across the board. But you may have also installed a metabolic handle with that CH2 in between the two heterocycles. Nonetheless, we prepared these compounds. We could actually start from intermediates from the previous synthesis and then just install a methyl, halogenate, and then displace. And there, we saw definitely a much better potency and spectrum than we had seen with the direct link pyrazole. But we also found that actually one of those intermediates, just the methyl intermediate, was actually even more potent and had a much better spectrum. And so there's a little serendipity here. And it wasn't really as obvious as how you'd make this transition. We started with the 6-membered ring, and you remove the carbonyl, and it was a weakly active compound. And even if you had kept that alkyl group on the top, it's actually still weakly active. If you drop the pyrazole with the alkyl group present, you're also weakly active. And we had actually switched to an aromatic ring in order to improve our chemistry, and that actually turned out to be a way to get us the extra activity, because it was only with the N-aryl ring that you ended up with highly potent compounds. And so at this point, we knew we had the photostability problem taken care of, we had excellent potency, excellent spectrum, and we were really excited to get it out to the field. And so we ran a field trial on Septoria tritici, our problem child, and we ran it against prothioconazole. And so prothioconazole at the time was the gold standard for control of Septoria in the field. Prothioconazole is run at 200 grams per hectare, and we ran all of our analogs at 300, 200, and 100. The test was curative, so that means that the plants were already infected prior to treatment. And what we saw with all analogs and at all rates was we were superior to the commercial standard. And so we were extremely excited about this. But like many things in crop protection, it parallels the story of Goldilocks and the Three Little Bears. And sometimes the porridge is too hot, and sometimes it's too cold. And so before we were too unstable, and now it actually turned out we were too stable. And so our soil half-lives were tremendously long, greater than 500 days. And we know from the European regulations that this just isn't going to cut it. There's too much risk of movement to groundwater. And so while we had solved a number of our problems, we ultimately had created another one, and this became yet another dead end for us. And so really thousands of compounds later and many different chemotypes, and I think probably every chemist in Discovery at Crop Protection were involved. And finally, a few years back, the opportunity to return to the field and come and see some of our field trials. And so normally we run our field trials in the center of a commercial field. And commercial growers normally spray maybe 2 to 3 times a year for wheat diseases. Well, that year, the Septoria tritici pressure had been completely unbelievable, nothing like that ever seen before. And so the grower that we were visiting had sprayed 7 times. We knew our protocols only had 2 applications, and so there's a current concern right off the bat that we might have lost the whole field season due to the really insane pressure. But when we came over the hill, in the sea of brown, what do you see? You see these nice green squares. And so what we saw up close were plants like this. And so in cereals production, and it's important to know that the top 2 leaves, the top leaf, which is known as the flag, and then the F-1 leaf, contribute between 70 to 80% of all the carbohydrates to the grain. And so if you're going to protect anything on the plant, you need to protect those 2 leaves. And so when you look at our control, and they're a little bit harder to see here, but you can see a lot of disease on the top 2 leaves. And all the other leaves are dead and senesced at this point. And so in the control scenario, likely there is not going to be really any more yield increase from this point on. And we're still 4 to 6 weeks from harvest. When you look at the DuPont experimental side, we're clean from top to bottom. Not only have we protected the yield-producing leaves, but also the leaves at the bottom, which produce inoculum for further infection. We started looking at spectrum. This is apple scab, which can occur on apples or on the leaves. And it's a similar story with yield. And when the leaves get infected, they'll start to curl up and eventually die. You lose yield on the apples. And then finally going out to grapes. This grape powdery mildew. And so that's the grayish purple fuzz on the grapes on the left, and on the right, nice and clean. And so at this point, we finally realized that we had actually accomplished all the goals we had set out. Our compound was exquisitely active. It was appropriately stable and appropriately unstable to make it through regulatory challenges. And so even after we had proved everything in the field, really the registration process for any agrochemical is very long. And it can take years until you finally prove the safety and prove the risk-reward benefit for your product. And so as a compound like this, which is broad spectrum, you might get involved in over 100 countries, get involved in hundreds of crops, which means hundreds of tests, both for efficacy and for toxicology, environmental fate, biological fate. But ultimately, you'll impact millions of farmers and billions of consumers. And that's, I think, what's really rewarding about working in crop protection. And so I'd like to take the time to accomplish folks who particularly contributed to the chemistry I talked about today. And so Jeff Long and Jim Bresnek were there to the bitter end and made key contributions that really got us to that final molecule. Also our associates, Cheryl Liberato, who's here today, Laura Geist, and Mike Mahaffrey. So these are the people who really to put those ideas together in the laboratory and get the molecules in the bottle. Tom Stevenson, who was my mentor when I started out at DuPont, got me started in the tubulin fungicides and made critical contributions to the pyrazidone chemistry. And then Tom Selby, who gave us a real jump start in the imidazole chemistry. And finally, George Lobb, who's also been an excellent mentor during my 10 years at DuPont. And then, as I mentioned, all those different attributes have a team behind them. And so things like environmental biological fate, led by Bob Dietrich, Mike Klaproth and Bob Foreman, who ran our discovery biology screens, and Benjamin Parotin, who's at our European Research Center, who ran our field programs, and then Jason Wall. As I mentioned, formulation is an extremely difficult situation, and he's led up our formulations effort to get us out to the marketplace. And then finally, this is a 10-year story, and there was a lot of times where we had to tell people we're going to start over, and we got a lot of patience from management, starting out with Mark Thompson and Mike Conding, and then finally it was Patty Parzielli and Bill Barnett who were there at the end when we moved out of discovery into development. And so really grateful to them. I'm really grateful to SCI and the Chemical Heritage Foundation for this opportunity today, and also grateful for DuPont for giving me the opportunity to do really exciting science with a big impact, and to let me to continue to have fun. And that's really what it's all about. And so with that, I'd be happy to answer any questions.
So do we have plenty of time for questions? I'll be running this, so put your hand up, and I'll do my best to bob and weave through the table, so just be patient for a second. So any questions for Drew?
Yeah, I was particularly interested in the grapes and the powdery mildew.
Mm-hmm.
And the main thing I was interested in was, and if you were here last night, you know, we always have good wines. So did you actually do a lot of work on both grapes to see what kind of wine they both made?
So that's actually a standard test for a grape fungicide. And so during the development process, you'll actually have to prove that there's no negative impact. And so they will grow the grapes under various protocols. They will make wine and they will taste it. DuPont actually has a high-level wine taster who's part of our R&D staff, and so it'll be his. It's a good job. Yeah. But that's something that I mean has been a pitfall in the past, and so you do actually have to really carry things out. Oh.
I noticed many of the compounds you showed, they had like an isoleucine or leucine type of side chain and a heterocyclic core, right? Could you comment on the mechanism? Do they?
So this area of tubulin fungicide actually binds to the vinblastine site, and so it's known in the literature from there. And if you're familiar with the structure of vinblastine, it's actually a very large, complex molecule. And it turns out these compounds bind to the same site, but with a much simpler structure. The side chain you're talking about, while sec-butyl in the case of the pyrazinones was the best, as you moved into some of the other chemistries, aromatic rings became better. And so while it seemed to be imperative in the beginning, as you went down the road, it turned to not be as important as you shuffled through different cores.
Andrew, very nice presentation. You mentioned it was a 10-year time period. How do you, what was the number one thing to keep your stakeholders engaged over that time period?
I don't know, I mean, the project died 1,000 deaths, and so, but it was always really the opportunity at the end. And so a broad spectrum fungicide can be particularly a novel mode of action, can be a once in a lifetime sort of opportunity. And so from a market standpoint, it's really huge. And then when you think about the growers and the consumers, it's imperative that we get these things. As I mentioned, when we ran against the commercial standard, I mean, it was performing reasonably well in that test, and just a few years later, you can go out to places where you could have it at 200 grams, 500 grams, or even a kilo, and it no longer works. And so that's a real catastrophe for that grower. If their whole harvest goes away because of resistance, I mean, this is, it's catastrophic to the person, to their family, and to their community. And so really the motivation is, how do you solve these problems? Of course, DuPont wants to make money, but also, there's that societal benefit that is also a huge driver for us.
This is two parts. You said that your compound binds to the vinblastine binding site. So the first part is how come it doesn't harm other kinds of organisms like vinblastine does? The second question is how long is it going to be before the fungi outsmart you and become resistant?
Yeah, well, so the first part, how does it not bind to other sites? And really, I mean, in reality, a lot of these molecules can. And so it all comes down to balancing the physical properties of the compound and, potentially installing a metabolic point that maybe mammals will metabolize, but not the fungus, or if you can make it so bioavailability is very poor. So essentially do the opposite of what pharmaceuticals would try to do, where they want to up oral availability, for instance. We actually try to dial it back. So there's poor absorption in mammals, poor absorptions in other organisms, and then hopefully high metabolism. Now, and how long will it take nature? Well, back in the 90s when, say, the strobilurin fungicides came out, people didn't really put a lot of thought into what nature was going to do, and you would send a fungicide out alone, so it wouldn't be sent out in a mixture with anything else. And things like the strobilurins really built up immense resistance in a fairly short period of time, maybe 5 to 7 years. Right now, what we try to do is actually pair each molecule with something with a similar spectrum of activity, but a different mode of action. And so the thinking is that by going out in combination, you could hold off nature, but eventually nature will win. And so it's good for the chemist because otherwise, a discovery would mean the end of your job. So, maybe you can push things longer than that. It's really hard to tell. A lot of it, comes down to what your mixing partner is and what happens in the environment. Because we've been surprised before with pathogens showing up, and there can be there are certain pathogens that will never get resistance to a strobilurin, and so those things remain to be seen.
All right, well then join me in thanking Drew for a wonderful talk. Don't forget the medal.
The goods here.
Yeah. And don't forget the lid. So this is great. Thank you for joining us for the lunch, for the presentation of the Moore Medal. You have about a 20-minute break to maybe stretch, go outside, and get a little bit of fresh air. We'll reconvene at 2:30 back up on the second floor for our second series of breakout sessions, responsible innovation, biomimicry, and industrial biotechnology. Stop by the desk if you have any questions. Otherwise, we'll see you in about 20 minutes. Thank you.